I'd like to introduce myself by telling you some of my childhood experiences and some of the ups and downs I encountered, followed by the area of research I'm interested in, as well as the work currently being done in my research group. Finally, I'd like to give my own perspective on where I'd like research to go in the field of autism as an autistic person as well as a researcher. At the time of my diagnosis, autism was a condition that little was known about in Ireland. During the middle of my second year, my parents were so concerned with my development and behaviour that I was referred to the Child Guidance Clinic in the Mater Hospital. Their causes of concern included absence of speech, total lack of communication skills, poor eye contact, unexplained tantrums, appearance of deafness, no interest in people, constant staring off into space as if I was in a hypnotic state, stereotypical behaviour with object flicking near the eye, unconventional play with toys such as turning toy vehicles upside down and spinning the wheels, poor eating habits. I would only eat food of one colour and shape, so that would be just about much you count on one hand. And repetitive actions such as trickling sand through my fingers and splashing water. It wasn't just lack of speech that concerned them, it was all these other oddities I displayed. My initial diagnosis was pervasive developmental delay, low IQ, and even hearing loss, which was diagnosed on account of my lack of response to others. I had grommets inserted into my ears, but they were removed as it became apparent that this made no difference. I was a non-verbal child who did not respond to verbal cues. It was during toilet training conversation at St. Paul's Hospital that autism was first mentioned to my mother, but when she tried to get more information, she was told that labels were not important. My mother felt that it was very important to her as she wanted to be proactive in getting help for me. I was brought to the Maudsley Hospital in London to see Dr. Rutter, a world leader in autism, to get a second opinion, and in the course of an afternoon consultation, I was definitively diagnosed with autism. So my fascination with water led me to, certain noises, smells and stimuli caused me great distress. I was so rigid in my routine that I would only eat cereal from the same bowl and sleep with the same duvet cover, which was washed, dried and put back on before my bedtime. Anything I ate got the nose test, so I would always be sniffing at food. If I didn't like the smell, I wouldn't touch it. I also had sleep disturbance for quite some period, and many of my activities were carried out in the early hours of the morning. I was like Houdini trying to climb out of my crib and escape, and which caused many sleepless hours for my mum. It was during these early morning excursions that my mum noticed, which led to one of the first breakthroughs. It was my fascination with water. When she hiding behind a door, she noticed me climbing up to the shelf, getting a glass, filling with water from the tap, putting it down over the plug hole. I had as I mentioned, I had a fascination with water, and that included flooding the kitchen sink, followed by the bathroom many years later. And she was monitoring this because she still saw the sink getting flooded without the plug hole there. So it was observing that when I used a glass filled with water up to a level where it wouldn't float when I started filling the sink, and once I got it to a level I was happy with, I was able to continue my activity of flooding the sink. It was from this that, as I mentioned, loving the water, that she had her breakthrough of using my fascination with water to learn about things, and also in helping acquiring language and play. So, as I mentioned, while it was a problem because as well as flooding this kitchen sink, I also flooded the bathroom, and there was one point where the bathroom flooded not only out into my brother's bedroom, which destroyed the carpet, and they had to get it replaced. So, in the earliest part, the most difficult part of my education was at the beginning, getting a start and finding a creche in play school that would accept me. I attended 6 different play schools. I became a serial preschooler, lasting from 1 week to a record of 8 weeks before I had to leave, as they did not appreciate my talents. Reasons were not sitting still or being unsociable to other kids who often poked me to hear funny noises. One preschool let me go because the assistant left and her replacement had no training with children with special needs, which was a shame because that school was very good. And you can see here, when I was in Cub Scouts, the first picture, you can see, this one here? So you can see here, the Cub Scouts, I was standing there in the corner, not being with everyone else. And in the lower part, you could see my mother here trying to keep me within the group so they could take a photo. And also, earlier on, I did want to engage with people, but I didn't really quite know how to do so. So you can see here in the background, there's me just climbing over a tree away from everyone else, me being a different sandbox from everybody. And here was when after the World Cup in 1994, when the Irish team had their homecoming, everyone was cheering and clapping for the team coming on the stage, and there is myself just not paying any attention, clapping at something else altogether, which was probably exciting because of all the noise. So it was during the earlier years that my mum decided to so thanks to a method used at home, including hours of one-to-one teaching, play therapy, language and behaviour management sessions, I finally got to start school. I would like to emphasise here that there were far less professionals with this kind of training 25 years ago, and that my mother had repeated whatever few sessions of speech therapy that we managed to get, as well as reading whatever she could, just improvising in general to help me with the obstacles I faced. And one example would be expanding my diet. She used bribery. Basically, she said if I ate a pea, I would get a hula hoop. She also got my siblings involved in playing games with food, so getting me to eat spaghetti. She got all the siblings, my brothers and sister, to start slurping the strands up and down like they're sucking up worms. And through a combination of all this, I was eventually able to gradually expand my diet. And now food is one of the simple pleasures I enjoy in life, particularly eating Chinese, Indian or Mexican food. Unfortunately, developing celiac disease later in life put a dampener on that, but still able to adjust all the same. And as well as my mum, I got a lot of help throughout the community. Here were some local neighbourhood boys that my mum often got by giving them £1 if they helped me play. Like say, here they had like the hoops, and they often play games with me, like, passing a ball or throwing hoops onto this little toy here. I also got very good help from my primary school, from my teachers in the Masher school, which I'll mention later on. My primary school teacher, such as my teacher and headmaster, as in a very small school when I became mainstreamed. He managed to include me in a lot of activities such as table quizzes and playing cricket, because I loved smacking the ball and running up and down, which I tended to do go by myself in the playground. And he realised, if I could harness that for playing cricket, and I really enjoyed that. Also, when I was In secondary school, I had a very good English teacher who helped teach me about not to always write factually, to use poetic license. And I'll tell you a funny story about that in my junior cert here later on. And then finally, even when I was in college, I had a very good occupational therapist who helped me with how time management, how to cope with stress and organizational skills. And it was also nice every 2 weeks to have a one-on-one with her to just say how things were going. And it was during the matter, it was in 1994, I had the big turning point. It was when during the school I started moving from echolaic speech, just parroting phrases and words which I had no understanding of, to actually doing basic communication. And it was when my mum recorded her notebook, which she used as a two-way communication between her and my teacher. They would write stuff, okay, this is what Jamie did at home, and then the teacher would use that to ask me what I would do, what I did at home for news time in the school, and then she would write notes down to give back to my mum to say this is what Jamie did in school. And it was through this notebook that around 1994 I started to respond and ask simple questions and commands. And I just wanted to highlight this picture because of this little point thing up here. Look who's talking now. But even though as I begun to speak and communicate, I was still lacking in a lot of social graces. So here was when I was doing my first first confession, where we were all going up to the priest to say, tell him of your sins. And so I went up and I was there for quite some time. My mother, who was in the audience, was getting very worried. He's been up there for a long time, about 10, 15 minutes. And it was just before she was going to go up to try and bring me down that I went back to my seat. And it was later afterwards she was talking to the priest and was like, oh, I'm sorry, Father, I hope he didn't bother you taking too much time. He goes like, oh no, he was very good. He confessed not only his sins, but the sins of your entire family, particularly of your father and your youngest brother. So I basically saved him a trip to the confession box, so there was always positive in that. Also when I was in the special unit in St. Patrick's, I was often a little bit used the wrong phrases for the wrong situation. So there was one substitute teacher we had there that I really liked, and when she was leaving, instead of saying goodbye, I said good riddance. Which was and then they had to explain to me, that that's a bad word. Like, I didn't understand that those words had different meaning, the words had different meanings. I just thought it was some way of saying goodbye or something like that. Also, never got the hang of lying. Because it can be quite common amongst those who are autistic, as we can be often quite direct with others. And this directness has often landed me in trouble. For example, I was at one of my brother's communions, and afterwards we're all having the tea and cake, and there was one of the priests there about to tuck into something really nice, and then I was there after reading a book about poison, so like food additives, toxins. I was just there going, oh, poison beware, and I started rattling off all of the colouring and additives in his cake, which I'd read earlier on. And my mother was like, oh, I'm so sorry. He kind of has a bit of speech and language difficulty, and the priest was like, certainly didn't ever sound like he had a problem to me. And also, even as I'm in junior cert, when I was doing the English exam, my mother asked me how I got on. And he said, did you answer all the questions? Like, oh no, I left some sections out. Like, oh, did you have a mid-attempt? No. And I don't, oh, why? And it was a section on poetry, where it was a poem by Patrick Cavan about where he wrote about his mother in memory when she passed away. And he goes, but you have a mother, you this would have had some meaning to you. And he was like, yes, Mum, but you're not dead. You see, this poem was about her dead mother, and I kind of took that very literally. There were also another question about where they were writing about the war, and I was, but you read up on the history of war. You love reading about the Second World War. And he goes, yes, but never fought in one. So it was, again, thanks to my Leaving Cert English teacher, he was able to kind of, and along with him, to teach about poetic license and to not always to write something factual. And it was through this help that I managed to surprise people. In my Leaving Cert, from not being able to speak, I managed to get an honours in English with an A1. And it was from there I went on to study genetics in Trinity College, where after, in this latter half, when I got to know more people in this much to our class, it was like one of the happiest times of my life, going out, socialising, having a few drinks. We even all went on a class holiday after we all graduated for a week over in Santa Ponsa, which was great fun. And from there I did my master's in Queen's, where I was living by myself for the first time, so I had to kind of make my way around, cook my own meals, and just make sure to hand in assignments on time and attend classes. And from there I am now currently doing my PhD in Galway, and now we'll start talking about the research I've been involved in. So, first, as Louise mentioned, many of the advancements we know from genetics has been through the use of large groups of individuals, and these would be biocollections. But what's a biocollection? So these would be, a biocollection is a repository that contains tissue from many people. So that could be blood, skin cells, anything, and also clinical data associated with the person. There are many advantages in collecting data this way. So you could have the samples have been well characterized, so they're very high quality and can be used for multiple studies by many researchers. This in turn can foster collaboration between researchers because they share all the data, it gets collected and it grows, and it opens up new questions that need to be answered. And importantly, it enables large-scale studies because there's a lot of the research in genetics requires huge numbers of people to even start finding something of interest. And having such large-scale studies can be very useful in this. But the question is, are such collections in existence for autism? And if so, have they contributed much to our understanding of autism spectrum disorder? And the answer is yes, and a lot. So I basically wrote a literature review that covered from the beginning of this millennium up until 2016, and I found 264 studies published from 5 collections. 4 of these are based on collecting blood samples, and one of them is based on actually collecting brain tissue to explore actual parts of the brain and to see how it's affected between those who are on the spectrum versus those who are not. In addition to that, there were 2 recent collections that have only been set up in the past few years, but they could be of significant interest further down the line. And they've contributed to some major studies. So they've identified some recurrent genes. This included neurexin-1, as Louise mentioned, a gene called PTN, which is involved in another significant syndrome called Prader-Willi. In addition to single genes, they've also found little deletions and duplications in regions of the chromosome, such as one such as 16p11.2, which is a very frequently occurring chromosomal anomaly that often coincides with some diagnosis of autism, and another one which is the 15q. And in addition to single genes, they've also found that you have de novo mutations, meaning that they're not transmitted by parents, but they often occur spontaneously in the child, and they're often significantly enriched. and they converge on sort of molecular pathways involved in synaptic transmission. This is basically the firing of signals between nerve cells. Chromatin, if you're wondering if DNA is the long strand which contains all the instructions as from the cookbook as Louise mentioned, the chromatin are the little balls that allow the strings to be wrapped up in really small spaces, because if you unfurl all the DNA, it can be 2 meters in length, and these little balls are allowed to be scrunched in to a tiny, tiny space. But these little balls also allow parts of the instructions to be sort of loosened up so they can be accessed to make the genes. There have also been emergence of discrete subtypes of autism linked to specific mutations in some genes. And there are just 3 here that have been found to be quite significant amongst the autistic population. And then the collection has also been involved in exploring factors, say, during pregnancy that could be related to autism risk, such as, say, immune responses to infection during pregnancy, or or stress. And even in the autistic people themselves, such as, say, looking at inflammatory markers, say, like if there's certain markers involved in immune responses that could be very different between autistics and non-autistics, and also the level of hormones, which can often be involved in brain development. So as for recent collections, now we're seeing collections, say, one which is the autism inpatient collection, which focuses on patients which are severe, on the very severe end of the spectrum, as in it can be very difficult to get them into regular clinics, so that often they're presented in hospital, where they found high levels of self-injurious behaviour and intellectual disability in this population, and they're hoping to start releasing genetic data where we'll be able to find out, say, what's different between those on the severe end of the spectrum compared to those who are, say, on the more milder or moderate ends of the spectrum. And then there's the Autism Spectrum Stem Cell Resource, which is set up in Orange County in California, where they have cell lines from over 200 donors. And the special thing they've done here is they've made using stem cells, basically I'll talk about these later on, but they have a whole collection of skin cells, blood cells, stem cells, and from the stem cells they're able to make all kinds of brain cells, such as the neural cells, which are involved in firing of signals, and glial cells are kind of the great support cells in the brain that have actually been found to have quite a few functions. So throughout this review, they all have advantages and disadvantages depending on what type of tissue you can collect. So for example, blood, it's fairly easy to collect from patients. You can store it in deep freeze, and you can use it for a lot of general genetics and even certain studies for looking at little compounds that could be circulated in the blood. But often the expression of genes in blood will be very different to that of the brain, which is what we'd be most interested in. You can also collect brain tissue, which obviously is the ideal thing to look at. So you can look at all the parts of the brain, say either how it develops or how it functions, and how it differs between autistic people and non-autistic people. But the problem is it's very hard to obtain. You only have a very small window of opportunity to get the brain when a person passes away and preserve it before the brain becomes useless, as little as 6 minutes essentially. And it also, depending on the part of the brain you're looking at, the results can differ in these studies. And often, because it's very difficult to get the brain tissue, the studies that they do are often very small. Not many people can be involved because, again, it's so difficult to get brain tissue. So here is where stem cells can be very useful. So stem cells are a type of cell that can be coaxed to produce any cell you'd like. That you can make them turn into heart, muscle tissue, brain tissue, tissue in the gut. You're only limited to what you want to explore in biology. And with the advantage of this, you can overcome the small sample limitation with trying to get actual brain tissue from deceased patients by making these cell lines, and you can scale up to do big studies where you can get a lot more insight. But there are some problems. You have the method on how to reprogram normal cells into stem cells. You don't want to do anything that could cause changes where if you're doing a study down the line, you think, oh, this could be really interesting to autism, but it turns out it could be something that actually happened in that reprogramming stage. So you have to be really careful there. And sometimes even still, particularly among some people on the spectrum, it can be very difficult to obtain a biopsy. You know, it's difficult enough to get blood from a person who's on the spectrum. Believe me, I would have had that same problem when I was much younger. So, and then, and so it was after a lot of work in work on this review that I finally got it published. So I just wanted to show this little picture of myself here with the paper. And this paper, by the way, is actually free, open access, so anyone can read it if they like. And if you want to take a note down, please feel free. So it was from after this review I realized there are actually hundreds of genes involved. You can't really say to autism is caused by any one gene. So I decided, well, on what systemic level are these genes involved in? So what I did was I used a database which has recorded all of the genes reported in autism, and I used publications where they had their own sort of scoring systems to assess how significant a particular gene could be related to autism spectrum disorder. So using the best 4 out of 5, I had narrowed it down from, say, tens of thousands of genes from each group to just 503 after overlapping all of them. Then I split them into 2 groups. So I have one group of genes which are known in an autism database, and there are others which, while not known to autism quite yet, they have still shown to be very significant, as in if they're mutated, something is very likely to go wrong, or often mutation could be associated with a certain condition. So I focused on the former list, and then I used another database which contains expression. I used a gene expression database where they took samples from many patients where they were wanting to explore the expression of various genes across all types of tissue. So the brain, the heart, the lung, the liver. I wanted to see, as well as the brain, were any of these genes expressed in other parts of the body. So I have this is what you call a heat map. Now a heat map is basically, you want to show like your signals of whether it's hot or cold. So in this case, all these little bars signify the expression level of a gene. So the red means it's very high, so hot, whereas the green is cool, cold. And here you can see in the brain, there's 2 blocks. So over here in this big block, you can see that not only are a load of these genes involved in the brain, but they're also expressed across the body here. Whereas you have another bunch of genes that are mainly just expressed in the brain itself. Now, when I got the results, it's all these tables that you push in. Have any of you used Microsoft Excel for like spreadsheets? Well, a lot of the results I got in analysis kind of looked like just all these tables. It's a bit boring, really. So what I did was I used some software to make nice, pretty diagrams like this. So this is showing all the relation of these genes and in what role they play in biology. So here in this big chunk is a lot of genes related to nervous system development, which is what has been kind of more or less found in autism spectrum disorder, as it is a neurodevelopmental condition. But you also find genes involved in up here, which is involved in heart tissue, funnily enough. And the interesting is, a lot of neurodevelopmental disorders, they found that there's some genes involved in neurodevelopmental disorders are also involved in congenital heart defects. And there are actually been reports of, say, the heart rates of people on the spectrum can be often irregular, say when they get stressed or excited. And so with all this, this is where I wanted to say about stem cells. Stem cells can be really useful in autism spectrum disorder because they can help us model the disease. You can actually grow these cells in a dish, and you're able to actually see how the cells grow and develop and compare between people on this between autistic samples derived from autistic people versus those who are not autistic. And even when the time I started my PhD, it was really, we were started with like 2D culture, so basically we're just growing just the little actual cells. Now we're actually able to grow mini brains, as in actual 3-dimensional segments of the brain, so you can actually look at, say, parts of the brain involved in memory, parts of the brain involved in coordination. Really exciting. I just wanted to show, this is the type of cell we're kind of trying to grow. This is what are called pyramidal neurons, basically because they're shaped triangularly like a pyramid. And these are the cells that we're looking at in the part of the brain called the cortex. And I just want to show a very neat picture where have the brain cells firing. So you can see that the cells are spontaneously giving off signals. And in the next slide, this is another one where we stimulate them. So they all light up like Christmas trees. See if there's one more little fire from them. No? Okay. It's not. So now, what I'd like to see in the future, well, through a combination of reading through the literature on my literature review and what I'm currently working on, I've noticed that there's quite a few comorbidities involved, basically conditions that often occur with autism. So as you all probably know, if you're parents, you'd have a lot of your kids probably might have gastrointestinal issues, like basically problems with the gut, or like, you know, having like a tummy aches or problems going to the toilet. And what I'd love to see is if there's some research, say, on these other systems, so like, you know, say all about the digestive system, so like the food pipe, stomach, pancreas, the intestines. I'd actually love to see some more work on the cardiovascular system, say like the heart and as well as the major veins and arteries, because there are some groups of people in the spectrum, very often the ones who are very severely affected or of intellectual disability, they often sometimes have heart issues or heart defects that often have to be corrected. And I would love also to use, in addition to say some animal models, so say looking at the condition with mice or rats, to also use stem cell technology to grow parts of these organs to get to look at to see what's going on, and to model the recorded mutations that could potentially have an effect not only in the brain, but in these other organs as well. And also I'd love to see more whole genome sequencing. In particular, I would love to see, there's technology now where you're able to sequence longer sections of DNA. So when you're doing sequencing, often the current technology for the past few years was you only have little fragments. It was trying to assemble a jigsaw. So if you any of you remember having to assemble a jigsaw, some of the big ones would often maybe have 100 to 1,000 pieces, and that took time. Think of it like that, except it involves millions of pieces. And often they use supercomputers to having to like use complex algorithms to reassemble those pieces in a way that might resemble what that cookbook could be. However, if you're able to have longer sections, you only have less pieces, and if you have less pieces, that takes less time to assemble the jigsaw. Or in the case, say, if you have just trying to reassemble a cookbook, and you only have little letters and maybe some sentences, having longer ones means you can maybe have whole pages or chapters to put the cookbook back together. So now you'll have a cookbook that has all the ingredients in the right place, and they may not have been put in the wrong place by accident, let's say, if that analogy makes any sense. And then as well as looking at the coding section of the genome, there's also the 99% that we don't know what happens. Originally, researchers thought that was junk and that it didn't do anything, but it turns out this section of the genome can actually be very important in controlling the expression of genes. So things like all these buttons and levers that determine how much or how little something can be expressed. So just an example of some of the other places I'd like to see would be the gastrointestinal tract. So one of the major type of nerve synapses that can be affected in autism is something called the glutamatergic synapse. This is a sort of, it sends off excitatory signals that makes, say, the brain do things. So this synapse is also found in all parts of the gut. So you can see there are parts which communicate between the brain and the gut. Some researchers are actually looking at this sort of what you call brain-gut axis, and they're actually looking to see if even the microorganisms in our gut could have an impact in autism. But I'd also love to see if any of the mutations found in the brain have an impact in these various receptors and synapses dotted around the gut as well. Because sometimes mutations here have been found to be associated with irritable bowel syndrome, secretion problems such as hypersecretion of gastric acid, or inflammatory bowel disease. And a lot of these conditions are disrupted in a lot of people on the spectrum. So could the same mutation, say, in the brain for some of these subjects, could they have a similar effect in the gut? I would like to see that. Another is metabolic conditions, where it's come recently through large studies across hospitals and medical care centres. Quite a few people on the spectrum have higher rates of, say, obesity, type 2 diabetes, hypertension, hyperlipidemia, so basically load of cholesterol in the blood. And very curious was non-alcoholic fatty liver disease. So often some of these conditions can be caused, say, if you're like a side effect of medication, or say if they have a poor diet. But what about the genes that have been found in autism could have other functions? So one example would be some genes that are involved in the release of, say, what are called neurotransmitters. These little chemicals that help carry the messages and across nerve cells. Some of the genes involved in the brain could also be involved in the pancreas. In particular, these three genes up here, they've been found to have a role also in the secretion in the pancreas. So I'd be really interested also to see if, to take a look at the metabolic aspect of autism as well, because that could be of significant concern on how to plan over an autistic person's lifespan and how to manage health. And then I would like to finish on just what lies ahead for us. So as mentioned about the AIMS project, there's been a growing demand for genetic testing across Europe on how to best help people on the spectrum and trying to figure out what possibly in genetics could be contributing to their condition. But the problem is, what kind of testing should we do? Should we just looking at chromosomal abnormalities? Should we do exome sequencing, where we're trying to look at the coding part of the genome? Should we look at the whole genome itself, look at everything? that could be a problem because there's it can be quite costly to do some of these. How do you plan regulations around this? So how do we inform clinicians on the best type of test to use to look at autism, or how should they even share this information, which could be the greatest concern, privacy? So in some countries, say in France, often after a genetic test, they could be legally compelled, I think it was either the doctor or the patients, to inform, say, other members of the family in case if they could be at risk from this condition. And then looking at autism across lifespan, how will autistics cope as we age? Could we be more susceptible or less susceptible to, say, some neurodegenerative disorders such as, say, Alzheimer's or Parkinson's? Because some of the genetics in autism can overlap with some of these other conditions, but we need to really look to see at those aspects. Is there an overlap, and if so, could there be an additional risk or no risk? This could be a very important issue. And likewise, as I mentioned, could we, as a result, be more or less vulnerable to age-related cognitive dysfunction? How will we be able to better manage ourselves as we get older? Will we be still able to live independently? Could we need assisted living solutions much earlier or later than, say, the general population? There are so many things we need to think about regarding this. So I'd like to finally give my acknowledgements. I would like to give my acknowledgements to Dr. Veronica and McInerney and Dr. Yanis Krasik, who helped us in collecting and processing the samples in Galway for the Nerixon project. I would like to thank all of the team on the Shen Lab group, both who've past and present. And I would like to thank my collaborators of the Literature Review, including Louise and Foss, and I'd like to thank you all for listening.